This is Miss Vance, and it's time for notes again. This is Unit 10, Part 2, about dihybrid crosses and the law of independent assortment. So you learned how to do Punnett squares in middle school, and you probably had some experience doing monohybrid crosses. You've had some experience doing some more of those, just to kind of review what you learned before. So now we're going to learn about something you haven't done before, and that's looking at two traits at one time, and that's called a dihybrid cross. So remember again, this is a little bit of review. Homologous chromosomes have the alleles for the characters. The locus is the specific location. Remember, the plural is loci. And that's where the location of a gene is on a chromosome. In homologous chromosomes, the alleles are at the same locus as you see here in the picture. Okay. Homozygous individuals have the same allele on both homologous chromosomes, and heterozygous ones have different ones. And so you see here just a picture. That's just a review, something that we learned about before. The law of independent assortment is something we talked about also in meiosis, the fact that the chromosomes are independent of each other, the way they separate. And so we're going to look at that in terms of genetics by looking at two characters at the same time. So dihybrid cross is when you have parental varieties that are different in two characters. And so here's what Mendel did when he was figuring this out. Okay, he had a parent generation that he crossed true breeding plants that had round yellow seeds with true breeding parents that had had wrinkled green seeds. When the F1 generation was produced and grew up into plants, they produced round yellow seeds. That's how he knew that round was dominant, yellow was recessive. Okay, when he let those self-fertilize in the F2 generation, there was something really striking that happened. Okay, 9/16 of them had round yellow seeds. That's both dominant characters. 3/16 had wrinkled yellow seeds, recessive for the first, dominant for the second. 3/16 had round green seeds, dominant for the first, recessive for the second. And 1/16 had wrinkled green seeds, recessive for both traits. So how in the world does that work? Okay, so here's a pictorial representation of what he did. Here's our parent generation. We have true breeding, round yellow, true breeding, wrinkled green. So they're going to the parents are going to have an allele for the color and the shape, right? So the R for the color, for the shape, the Y for the color. Same thing in the recessive. And so the F1 generation is going to inherit 2 R's for the shape and 2 Y's for the color. And so in the case of these peas, we ended up with all round yellow peas. They're round and yellow because those are the dominant traits. All right. Now, if those things were inherited together, if they depended on each other to be inherited, if they could only be inherited together, what you would have is half of the sperm would have both dominant alleles, and half would have both recessive ones. Same thing for the female parent, the egg. Then you'd end up with 3/4 round and yellow and 1/4 green and wrinkled. That's not what happened. That would be if they depended on each other. So that indicated that they were independent of each other. So in which case they were inherited separately. And so here's what you have. You have these different combinations that can have. You can have both dominant alleles, recessive for the first, dominant for the second, dominant for the first, recessive for the second, and recessive for both. And you can have those combinations from both parents. When you put that in a bigger Punnett square, since you now have 4 possibilities of the different kinds of gametes, you're going to end up with these different combinations. And every time this cross was done, he came up with similar results, numbers that worked out to be the same proportion. And he was dealing with not just 16 plants, but he was dealing with 1,000 plants or more. Okay. But every time, about 9/16 of them were yellow round, about 3/16 were green round, about 3/6 were yellow wrinkled, and about 1/16 were green and wrinkled. Okay, And this is what he came up with. So that we call that a 9:3 to 3:1 ratio. That's a phenotype ratio. The genotypes are a little bit more complicated, and we're not going to worry too much about figuring that out. But understanding how the genotype ratio is going to work is going to be our main focus. Okay. So he had to figure out a way to explain that, because now there were non-parental combinations of traits. Okay. So his suggestion was that the inheritance of one characteristic has no effect on the inheritance of another. And this suggests also that the dihybrid cross is the equivalent to two monohybrid crosses, and he called it the law of independent assortment. Okay, and we talked about this in meiosis. We talked about the fact that the chromosomes are independent of each other in terms of which one goes which direction during meiosis. We also call that independent assortment. So these are very closely related hypotheses or laws that work together. So let's look at independent assortment as applies to these two characteristics in Labrador retrievers. We're going to talk about black versus chocolate color, and normal vision versus progressive retinal atrophy, which is an inherited blindness condition that is commonly found in Labrador retrievers. Okay, so here we have two dogs that we're going to cross. We have one that has a black coat with normal vision, so we know there's a dominant allele for each of those traits there. Here's one that's blind, has a black coat, and is blind, so we know that it's dominant for the first and then homozygous recessive for that second trait. Here's one with a chocolate coat with normal vision, so we know they're homozygous recessive for the coat color, and they have at least one dominant allele for normal vision. And then a chocolate coat and blind with progressive retinal atrophy is homozygous recessive for both traits. All right, so if you made it double heterozygotes, black coat, normal vision, and knew that they were heterozygous for those traits, then you would expect in their offspring, okay, over the period of time, they would not have 16, usually have 16 puppies at one time, and they certainly wouldn't have all 16 like this. Okay, this is just proportion over the period of time that you would breed these dogs together. About 9/16 of them would have black coat with normal vision. About 3/16 with black coat and blind. About 3/16 chocolate coat with normal vision, and about 1/16 chocolate coat and blind. Okay, and so that's the way it works out. And again, it's not every time there's a fertilization event, there's a brand new Punnett square, and the probabilities start over again every single time. So it's not like the totality of all their offspring would work out with these numbers. It's just that every single time the probability is the same, and so you're going to end up overall with numbers that are going to be pretty similar to what you predicted. All right, so how do you go about doing this? So this is something that you're expected to learn how to do in pre-AP in biology, but especially in pre-AP biology. Okay, you have to figure out the different combinations of gametes that can be produced by parents. And we use a method that you've learned in math class called FOIL, first, outside, inside, last. And so if you'll look at the little GIF here in the upper left-hand corner, this shows how you go about figuring this out. All right, you're going to take the first letter of each pair, the outside letter of each pair, the inside letter of each pair, and the last letter of each pair, and put those together. So you can see how that works. So I'm just going to make up some hypothetical alleles just because it's just easier to work with some letters. Okay, we're not going to use H and S. Okay, we're just going to use A and B. All right, so if we have an individual that is homozygous dominant for trait A and trait B, and one that's homozygous recessive for both traits, then we're going to produce offspring that are heterozygous for both traits. All right, so how do we figure out the alleles? All right, what are we going to get when we cross those two? So our possible alleles, again, if we take the first one that's capital for both or dominant for both, the outside ones, which are capital and lowercase, capital for the first, lowercase for the second, the inside ones, lowercase then capital, and the last ones, lowercase for both, we're going to get those four combinations of those two traits that are possible in the gametes. Same thing for the other parent. When we do this cross, we're going to put both letters in the box. So you're going to have two letters for each row and column on the outside, and you have 4 letters in each box, okay? And you're going to keep the A's together and the B's together. You're going to keep the capitals first and the lowercase second of each one, okay? So our first one here is capital A, capital A, capital B, and then we have big A, big A, big B, little b, big A, little a, big B, big B, big A, little a, big B, little b, okay? And we'll end up, for each one of these possible combinations, we end up with those different combinations there. Now there are about 9, I think, different genotype ratios. We're not going to worry too much about the genotype ratios, but the phenotype ratios are really important, and you'll have a 9:3 to 3:1 ratio that you'll get in the phenotype. So you'll have some more problems to work. These are dihybrid crosses, and you'll be looking at the different things. Now, one thing I want to point out to you, when the parents aren't a dihybrid like you see here, maybe one parent has this type of genotype and one parent has this type of genotype, then you don't necessarily have to do all 16 squares in your Punnett square. If this parent can only produce one kind of gametes, then there's no reason to repeat the same thing 4 times. So if for instance, if this is the only kind of gamete that this parent can make, but the other parent can make all 4, then just do the one row, because you're going to get the same proportion each time. So be reasonable. Don't think that I'm expecting you to do every single square every single time, because it just isn't necessary if you have only 1 or 2 types of gametes for one parent, as opposed to 3 or 4 different kinds for the other parent. Just do what's needed to be done in those worksheets. So work your way through the problems, submit them on Schoology. If you have questions, send me a message on Schoology, and I'll be glad to help you. And that's the end of part 2.